Here we see variations on gene exchange, all from the perspective of recombination. With genetic recombination, you're basically bringing DNA from two different organisms into the same cytoplasm or nucleus, and that's what's being shown here. The source of this DNA could be a gamete, it could be a virus, it could be DNA that's just floating in the extracellular environment. When you bring the DNA from two different organisms into the same cytoplasm, they have some potential to interact, but they don't have to interact. In conjugation, you're bringing in loci that comes from two different sources, and they don't recombine further beyond just having the two DNAs being brought together into the same cell. When you have molecular recombination, what you have are the DNAs that are brought together from two different parents in the course of gene exchange. What you have those DNAs exchanging segments, and as a consequence, the DNA itself, the chromosomes themselves, or the plasmid itself becomes a recombinant molecule. And then there's a third possibility where you have, in fact, the DNA being brought together into the same cell, but the DNA remains encapsulated inside of the cell, in this case as a endosymbiont. So you have the DNA associated with the host cell, the larger cell. This could be, and typically is, found in the nucleus of that cell. And then you also have DNA that's associated with an endosymbiont, such as a mitochondrion or a plastid. You can see that there's a great deal of similarity between what's going on here and what's going on up there. And indeed, you can have this DNA moving first into the cytoplasm and then into the nucleus and then integrating into the nuclear genome, and you end up with molecular recombination as well.